Okay. Assalamualaikum and good day everyone. So we are group 8 consisting of 5 members. Me, FARAHAH binti ZUHAIRI, AKHMAL DANIAL, NURAINI MOKHTAR, EIZZATI and NISHA AZURIN. So today we would like to explain our mind map based on an article named Cell Sources for Cultivated Meat, Applications and Considerations Throughout the Production Workflow. So we divided this article into 5 parts, which are cell types that will be presented by Nuraini, cellular considerations for scale up by IRDEENA, culture medium considerations by me, bioreactor considerations by AKHMAL, for considerations by NISHA. But before that, I will briefly explain what cultivated meat is and the processes involved. So basically, cultivated meat is the in vitro production of meat from animal cells. So the steps consist of sorting from animal cells. We basically obtain the cells from desired animals. Then the cells will differentiate into skeletal muscle that will then sort into the cell types of interest. The step will be followed by the bioreactor expansion that will then seed the cells in scaffold. And lastly, the cultivated meat will be produced. Okay, we will go on with the steps, which with the part of cell type will be presented by Nuraini.
Okay, thank you, Farahah. Okay, let's start the presentation with the cell types that is used for the cultivated meat. So usually we will be using the stem cells because they are the strongest candidate to use as the starting cell source. And there are two main types of the stem cell, which are the adult stem cell, which they possess the proliferative capacity, and pluripotent stem cell, which they also involved in the differentiation potential necessary. Okay, the first one, which is the adult stem cell, it is a more commonly employed cell source. It is an undifferentiated progenitor cells that reside in specific organs and tissue, and it is also multipotent, as these cells have the ability to differentiate into a select number of cell types. And there are three major progenitor are present in the muscle tissue environment, which include the muscle satellite cell, mesenchymal stem or stromal cells, MSC, and fibrio or adipogenetic progenitor, FAPS. All of these, they have the ability to differentiate into one or more key matured cell types, namely as skeletal myocyte, adipocyte, chondrocytes, and fibroblasts. So let's move on into the first one, which is the muscle. The muscle satellite cells is a muscle resident stem cells that located under the basement membrane of muscle fibers, and they can differentiate into myocytes, which in the end it will forming multinucleated myotubes that pack into myofibers. The next one is the MSC, which it have the capability of differentiating into adipocyte, chondrocytes, and fibroblasts. The next one is the FAP, which we consider as a separate mesenchymal cells population that resides in the interstitial space of the skeletal. Okay, next, the pluripotent stem cells. They are highly proliferative in culture and able to differentiate into any cell types that present in the three primary germ layers, such as the mesoderm, endoderm, and ectoderm. So there are two types of stem cells that are derived from the pluripotent stem cells, which the first one is the embryonic stem cells, or we call as ESC. For the ESC, it can divide indefinitely in vitro while maintaining the capacity to generate all the cell types of an adult organism. But it is often difficult to obtain due to the short lifespan of the blastocyst. And the last one is the induced pluripotent stem, or in short is IPSCS. So for this one, it is generated by cell reprogramming of somatic cells via the induction of genes associated with the pluripotency. But it is also less developed due to the require of additional studies to optimize the efficiency of the cell reprogramming protocol for different animal species. So that's all from me. I'm going to pass the presentation to Irdina.
Thank you, Nuraini. So I will continue with cellular considerations for scale up. There are two approaches to achieve scale up, which establish new pluripotent cell lines from key livestock species and improve proliferative capacity of adult stem cells. So for the first approach, the first important step to develop cell lines from livestock species is by identifying species differences in pluripotency factors. This is because base pair sequence and identity of specific factors that regulate cell pluripotency may vary depending on the species, which sometimes may or may not express in human pluripotent stem cells. Next, for the next, the first important aspect under the second approach is choose the cell type that have proliferative capacity. So this can be done by inhibit proteins that involve in differentiation, or by using novel genetic engineering techniques to target the genes that involve in proliferation. The next important consideration is differentiation of starting cell source into desired mature cell types with high selectivity and yield. And the final aspect that we need to consider regarding scalability of cell sources is level of structure in the final cultivated meat product. So is it likely that non-structured meat like minced meat more easily scalable and economically feasible to develop in short term rather than structured meat cuts? And that's all from me. So I will pass the presentation to Farahah.
All right, thank you, Dina. To the culture medium considerations. So basically, it is important to supplement the proper selection of nutrients and growth factors into the culture medium for supporting cell proliferation and differentiation. So commonly, basal medium formation like DMEM, L-glutamine, non-essential amino acid, and FGF2 will be included for adult stem cells. FGF2, which is the additional growth factors, is also included to improve growth or direct cell behavior. Then animal serum, which is the fetal bovine serum, FBS, contains a variety of growth factors, nutrients, and proteins for cell growth and addition. However, it got some issues like batch-to-batch variability in serum production and may be risky to be contaminated by viruses and prions. So the alternative to overcome these issues are by using serum-free supplements and using growth factors expressed as recombinant proteins. Apart from that, the cost is also one of the vital parts in this study. So in reducing the cost, we could recycle the conditioned medium during bioreactor expansion and also use serum-free supplements. So that is all for my part. Next, AKHMAL will explain about the bioreactor considerations.
Okay, thank you. So my part, I will talk about bioreactor consideration. So bioreactor is a cell expansion technology that provides stimuli to achieve scale up cell sources for cultivated meat. The mode of medium introduction into main vessel of bioreactor and the mechanism to achieve mixing of contents are important aspects to consider the types of bioreactor. So there are 3 types of bioreactor that are classified based on the mode of medium introduction into the main vessel of bioreactor, which are batch, and continuous. A batch bioreactor contains a fixed volume of medium and functions by growing cells to the maximum density, then removing the cells for use or transfer into another bioreactor with larger vessel size. A fed batch reactor, a bioreactor contains an inlet channel to feed fresh medium to the cells at set time scale selected to maximize proliferation, proliferation, and it also characterized by having increased volume over time, since it does not possess an outlet channel to remove conditioned medium and cellular product that accumulate during culture. While in continuous bioreactor culture, fresh medium is added to the main vessel and an optimized flow rate, while conditioned medium and cellular product are simultaneously removed. So the types of bioreactor based on the mechanism to achieve mixing the content are mechanical bioreactor, hydraulic bioreactor, and pneumatic bioreactor. The type of mechanical bioreactor are stirred tank bioreactor that which use an impeller to steer the content of bioreactor to create convective flow and aid in nutrient circulation and diffusion in the bioreactor around its central axis to dynamically culture the vessel content in suspension. Next, hydraulic bioreactor or hollow fiber bioreactor involve seeding the cells in a matrix with porous hollow fibers to allow cells to adhere to the hollow fiber surface where the medium may also circulate. And lastly, the pneumatic bioreactor, which accomplishes mixing via gas purging. So I think that's all from me. I'll pass to the next person to continue this presentation.
Right, thank you, Akmal. Next is biological scaffold consideration. Okay, tissue scaffold is used to support cell differentiation and tissue formation during cultivated meat production. For cultivated meat, scaffolds enable differentiation of stem cells via anchorage and to influence the shape and cellular arrangement of the final product, which is the meat. So one frequently employed scaffold type is microcarriers, which are micro-scale spherical beads that can be created from natural or synthetic biomaterials. It has a very large surface area to volume ratio that aids in scaling up the cell numbers, as it enables a large density of cells to adhere to the microcarrier. It also improves taste, color, and also the texture. So next is 3D bioprinting. So it is which is to create scaffolds for cultivated meat production. It functions by using computer-aided design models, which instruct the 3D bioprinter where to deposit a bioink. So there are three types of 3D bioprinters. Firstly is inject. It uses a thermal or piezoelectric system to dispense bioink from their nozzles. It is relatively cheap and very precise and unlikely to damage the cell. Next is laser. Laser bioprinter, which it utilizes a laser in conjunction with an optical lens to print a micro scale droplet. It has a nozzle free, which gives a larger viscosities. Finally is extrusion bioprinter, which allow very high viscosity and viscosity bioink to be used and result in a scaffold with strong mechanical properties. That's all from me. Thank you.
Right. I think that's all for today's presentation from the floor. With that, thank you so much, everyone, for this fruitful explanation and mind map done by everyone.
Right. Thank you.
Thank you, everyone.